Well, obviously we have a rapist in Lincoln Park.
Nucleus, a specialized, usually spherical mass of protoplasm encased in a double membrane and found in most living eukaryotic cells, directing their growth, metabolism, and reproduction, and functioning in the transmission of genic characters. In the nucleus, transpiration, the first step of protein synthesis, occurs. In the nucleolus of the cell, there is a double-stranded piece of DNA. Beginning at the tatabox, the DNA helicase serves to split the two strands of DNA apart. This is a single-stranded mRNA. It has a 5-carbon sugar called ribose and a base pairing rule of adenine with uracil and guanine with cytosine. The mRNA carries the genetic information from the nucleolus through the cytoplasm and to the ribosome. The mRNA will attach to the sense strand. The strand of DNA that the mRNA does not attach to is the missense strand. RNA polymerase will bind the mRNA to the DNA sense strand. The sense strand of DNA will separate from the messenger RNA. The SNRPs will cut out the intron, which are pieces of the gene that are not relevantly expressed, leaving behind exons, which are the expressed portions of the gene. The spliceosome brings the exons back together. The 5' cap attaches to the 5' end of the mRNA, and the poly A tail attaches to the 3' end. They will protect the mRNA as it travels through the cytoplasm to the ribosome.
On the road again, going places that I've never been, seeing things that I may never see again. I can't wait to get on the road again.
Now we're at the ribosome, a tiny, somewhat mitten-shaped organelle occurring in great numbers in the cell cytoplasm, either freely, in small clusters, or attached to the outer surfaces of endoplasmic reticula, and functioning as the site of protein manufacture. In the ribosome, translation, the final step of protein synthesis, occurs.
Transfer RNA, or tRNA, carries amino acids to the ribosome. The first tRNA will bring an amino acid to the A site. The anticodon of the tRNA will match with the codon of the ribosome. Once attached, the tRNA is transferred along with its amino acids to the P site. Another tRNA attaches to the now empty A site. The amino acid from the tRNA at the P site transfers to the amino acid at the A site, forming a peptide bond. As the tRNA at the P site moves to the E site, the tRNA at the A site moves to the P site, and a new tRNA with another amino acid moves to the A site. The tRNA at the E site is now ejected into the cytoplasm. This process continues until they reach a stop codon. At this point, the polypeptide, aka protein, is released into the cytoplasm of the cell. A codon can only code for one amino acid, but many codons can code for the same amino acid. This shows redundancy with no ambiguity. The final product of protein synthesis is a protein.
The process repeats itself to make many different proteins for the cell.
R-I-B-O-S-O-M-E, a ribosome where tRNA carries an amino acid at A site, P site, E site. That's where it happens. What? We need a protein. A site, the codon matches the anticodon. The tRNA brings an amino acid. P site, the next step. The synthesis don't stop. No. Because at the P site we make peptide bonds with amino acids. I said these will make protein for our cells, almost ready to happen. The RNA is done. Inside our plasma flung. The peptide bonds are strong. Beside we have our thought, protein is complete.
The set. God.